Just share my screen now. Are you able to see my screen?
Yes, you can just put it in presentation mode and then all good. There we go, perfect.
Okay, so good afternoon, everyone. My name is Jessie Kanyerere Amici. I'll be presenting today part of my PhD work. I'm with University of the Western Cape, and those are my supervisors. And as Natalie introduced, I'll be talking about understanding the occurrence of selected micropollutants from land to sea continuum, a case study of selected coastal areas of South Africa. So it will just be a bit different from the machine learning, that the data science visualization that we have been hearing from Latispangi. So first of all, what are micropollutants? So micropollutants are substances that are released into the environment in a small concentration as byproducts of various human activities. They are invisible and they are found in low levels in water, air, sediments, as well as aquatic organisms. So examples of micropollutants include pharmaceuticals, pesticides, industrial chemicals, and we have like personal care products. And these are from different sources. We talk of hospitals, domestic wastewaters, landfills, as well as agricultural runoff. So for this presentation, I'll be focusing on one part of my research. So I am looking at micropollutants, which is pesticides and pharmaceuticals. So I'll be talking much on the pharmaceuticals because that's the part of my research that I am currently almost done with my analysis. So a little background of my study. Worldly, we all know that we need pesticides, and most especially in agriculture. In terms of international demand for food and services, drives environmental footprints for the use of pesticides. And pesticides have been designed to prevent these yield losses, but also they have also given a rise of a new problem. So on the screen above, you can see that we have a graph that is showing us how much pesticides we are using currently in the world. That's the top diagram. And then the below diagram is showing us how the pesticides increasing consumption have been from 1990 to 2000 to 2021. Apart from that, when we look at pharmaceuticals, with aging and rising rates of chronic diseases, with pain management, there is a need for pharmaceuticals in our environment and in the whole world as say. However, these pharmaceuticals, they are behavioral issues. There is overprescription, which can lead to addiction and social problems. And there's lack of access to alternative treatments of these pains. Therefore, people go on, and now we have this overprescription of medication. And there's improper disposal of these pharmaceuticals in our environment. We see that people are just trashing them anyhow. There's no well understanding of how do we dispose these chemicals so that they don't have the impact on the environment. At the same time, the poor wastewater treatment plants that we have that are not able to break down these compounds. Because of that, they are not able to be broken down. These compounds are now sent to the environment, which leads to going into the ocean or the water that we're drinking, as well as the sediments. So on the above, you can also see that we have a graph that is showing us the use of the rivers that have been contaminated with pharmaceuticals, the global pharmaceutical contamination. And from this, we can see that sub-Saharan Africa is the leading continent in terms of this pollution. And down here, we can see the amount of pharmaceuticals that have been detected in surface water, groundwater, tap water, as well as our drinking water. So from here, we can see that these chemicals are not leaving any environment, and they are found in each and every environment. Therefore, that's why there's a need for us to understand this study and what are the impacts of these micropollutants. So The aim of my study is to understand the source to sink of the distribution of the selected micropollutants along the coastal areas of the Western Cape, because they are limited studies in the Western Cape and in the coastal areas. So in order for me to achieve this aim, I have some objectives, which I have to identify and characterize some sources of micropollutants. And then after that, we have to assess the distribution of these micropollutants in terms of transport mechanism and identifying them from land to sea. And then after that, I have to compare on a regional scale and the global trends of these micropollutants. So here is just my preliminary map that I am working on. You can see the shapefile of the Western Cape. And we also have different types of fish that we are going to analyze for these micropollutants. So far, as I indicated in the beginning, I have done some analysis on the pharmaceuticals and on the fish as my sink that is going to show me to see if these pollutants are indeed going into the ocean and at what concentration. So we have different types of fish. We have the deep ocean and the pelagic fish. And we also have the catchments, where we are now looking at the agriculture pollution, which is focusing on the pesticides. And we want to see how these pesticides move from the upper catchment going down to the ocean. So we also include groundwater, we also include sediments, and we are also looking at rivers. So based on the methodology, I will focus more on how I prepared my fish samples and how I was able to get some preliminary results from this. So the fish samples that we're using, we got it from the DFFE here in the Western Cape. So when we got this fish, it was fresh. So we had to freeze dry it. So when we're doing freeze drying, we are trying to remove the moisture content from the fish so that we just have the flesh in it. After that, we grinded the fish. And then from that powder, grinded powder, we now weighed about 1.0050 to 1.0070 of the mass. From there now we did an ultrasonic extraction, where it's involving that I have to add methanol to the fish, and then I will ultrasonic it for 30 minutes, and the water should be at 30 degrees. And then after that process, we'll take the fish samples to a centrifuge where it's going to spin like for 10 minutes so that the salt can sink down so that we can be able to extract the solvent from it. After that, that process is done like 3 times, because at the end of the day, we need like 9 mils of the solvent from the fish that we're extracting. From there, now we're going to take that fish and pass it through the cartridge. So the reason why we're passing it through the cartridge is for us to be able to trap the contaminants in that cartridge foam. So before we do that, we activate our cartridge with methanol and ultrasonic, micro millipore water. After we have done that, we pass in the 9 mils of the sonic extraction that we have taken from the fish, and then we pass it through the SPE at a very low pressure so that we don't break the cartridge. After we have done that, we dry the cartridge for 3 days, or we can use nitrogen drying for almost 30 minutes. After we have dried it, we elute it with methanol so that we are collecting the trapped contaminants that we assume we have collected it during the SPE process. After that, we now take the dried cartridge and dry it under nitrogen. Then we take it to CAF, which is Central Analytical Facility in Stellenbosch, where now we analyze the samples. But how are we going to analyze this? We have different standards and internal standards that we are going to spike our samples with so that it can be able to be detected on the machine. From there, we are going to get the results and work on the results. So far from the preliminary results that we have had, we have noticed that pharmaceuticals in the fish are there. However, they are not in that big concentration, because as it's a continued analysis. So for now, I won't be able to give in the exact amount on how much we found those pharmaceuticals in the fish. But so far, the common pharmaceuticals that have been detected in this fish is acetaminophen, we have sulfamethoxazole, triclosan, and ropinvir. And if you can see this, we have some painkillers, we have the prescribed drug, we have some contraceptives, and these are the fish that we have identified both in the deep ocean fish and the pelagic fish, which is kingklip, monk, anchovy, and red eye. So as the research is still ongoing, and we are still also waiting for some results to come in, and if these results come in, I will be able to present the full processed results in other platform of presentation. But for now, this is all that I had. Thank you so much for the opportunity.
Thanks so much, Jessie. That's a very interesting project you have going on. I'll be interested to see the results when you have some more, or if it's publishable at the time. I will open the floor to any questions. Please raise your hand if you have any. Or type it in the chat box.